A very good evening to all our attendees from India, of course, because our speakers are they are from US and it's morning there. I am Charu Seth from Fast India, and I'm joined by my colleague Utsav Thapliyal, who's here managing this webinar behind the scenes. We welcome you to the discussion on seeing science through lenses. It's a webinar on science photography as part of the India Science Festival. You may be aware of one of our flagship competitions called Science in Focus, which is a science photography competition. In case you haven't submitted your entries yet, please do so by 27th October and get a chance to win exciting prizes. Plus, the top 30 entries will be displayed at a dazzling photo exhibition at the festival on the 20th and 21st of January 2024 in Pune. My colleague Utsa will share the details about this competition in the chat window. Now, if you need inspiration on submitting the entries for this competition, you are at the right place, my friends, because we have here the experts of science photography, Michael Perez and Felice Frankel. Now, before I introduce the speakers, I'd like to tell you briefly about Fast India and the India Science Festival. Now, FAST India, or the Foundation for Advancing Science and Technology, is a non-governmental, not-for-profit think tank working towards enhancing the science and technology ecosystem in our country through various interventions, namely government support, institutional strengthening, corporate sector engagement, and science communication. India Science Festival is the flagship program of FAST India, which comes under the science communication vertical. And it has already emerged as the largest non-governmental platform for public celebration of science and technology. Our aim is to bring all the actors of the scientific ecosystem together for a much needed dialogue, break the silos, and bring science and technology to the forefront of the public imagination in the country. Now, talking about connecting science and society, I'm sure photography is one of the coolest mediums to communicate science, and you can all agree on that. I'm pretty sure about it. Now, for this webinar, we are honored to have Felice and Michael with us. Now, before I introduce the speakers, I would like to give you quick housekeeping instructions. You're requested to keep yourselves on mute and please have your videos on if possible through the course of this session. In case of any technical issues, please rejoin using the same Zoom meeting link. Attendees can drop their questions in the chat box and we would be happy to take them if the time permits. Irrelevant questions will not be entertained. To open the chat box, click on the three dots in the Zoom window taskbar and click on the option that mentions chat. The session will be recorded and the recording will be shared with all the attendees in due course. And the moderator will ask questions to the speaker wherever necessary. Okay, now I'm sure you want me to shut up, but let me just introduce my speakers first, and then I'll shut up and I promise that these speakers will make this session as engaging as possible. Felice Frankel is a science photographer and a researcher at the Massachusetts Institute of Technology since 1994. Felice is a fellow of the American Association for the Advancement of Science. He's previously worked in Harvard University's Faculty of Arts and Sciences and at Harvard Medical School's Department of Systems Biology. In 2001, she founded the Image and Meaning workshops and conferences whose purpose was to develop new approaches to promote the public understanding of science through visual expression. So, you know, Felice is all big on science communication. Her images have appeared in very reputed journals and outlets such as Nature, Science, JACS, PNAs, and so on. She has also been profiled in the New York Times, Wired, Life, Boston, among many others. The list is very long. She has received multiple honors and awards. Some of them are Progress Award from the Photographic Society of America, Leonard Nilsson Award for Scientific Photography, Chancellor's Distinguished Visiting Fellow in the Arts and Sciences at UC Irvine. So these are only a few of the list. You know, Felice and Michael had worked together before, and they are really glad to be doing this webinar together for our Indian audience. Michael Perez is a professor of biomedical photographic communications at Rochester Institute of Technology, where he teaches courses on applied scientific photography and visual communication. He loves using the classroom as a learning laboratory, and the core principle of his teaching is experiential learning. A recurring theme of his work for decades has been photographing things which are barely visible to the naked eye. His work has been featured by CNN, Time, Popular Photography, Mashable, Colors Magazine, and BuzzFeed. He enjoys sharing science photographs in non-scientific environments and is pretty active on Instagram. You can check out his fascinating work there. During his career, he has been recognized for several professional achievements, such as RIT Eisenhart Award for Outstanding Teaching, the College of Art and Design Gittner Prize for Outstanding Achievement in Graphic Arts, Biocommunications Association Schmidt Award presented for Lifetime Achievement in Life Sciences Communications, just to name a few. He has also produced and published 11 books. Now, fun fact about Michael, he began taking pictures in 1973 when he bought his first camera with money earned from bagging groceries at a local supermarket. And his life changed profoundly after discovering photography. Now, let me give the floor to Michael. Michael, we see how you got into photography. How about science photography in particular? And we would also love to hear interesting stories behind some of the photos that you cherish. Over to you, Michael.
Thank you, Sharu. Nice to be here. I have a little cold, so please bear with me as I work through that. So when Felice and I and Sharu spoke last week about how we would use our session, what we discussed that I had to go first. I don't know why Felice decided that, but that's what she decided. So I'm going first. I have 15 minutes to give you a little bit of insights and some of my history. And then I'll turn the platform over to Felice, who will do the same. And then the last 30 minutes of our time together, we can talk about some of the making of pictures. So I thought in my 15 minutes, I'd talk a little bit about this whole notion of images in science and the kind of concept that I create science facts, not science fiction, that my pictures are truthful, even though it sometimes may seem like their interpretations or beautiful. In science, people use different practices than maybe photojournalism or advertising. So the science photographer is bound by scientific methods. And I use different tools than the photojournalist or the advertising photographer or the artist. So in my world, I use infrared and I use microscopes and I use close-up lenses and fiber optic lighting. I have to photograph in ways that are accepted by my industry, because if I do too much interpretation or I interject a bias, it might no longer be a truthful image. So it's very important that I standardize my methods and photograph in the same way and do it in a way that industry is going to accept it. In science, my camera is not necessarily a camera as much as it's a data collection tool. It's a tool that records behaviors or situations that might be invisible. And so the camera collects the data of an experiment or an object behavior or characteristics of things. So science pictures become visual information or visual data about something that live to tell the story tomorrow. Photography and science will describe things that are very difficult to do in words. I ask you just for a minute to consider how you would describe the color blue in words. How accurate could you use, and how many words would it take to describe the color blue? I would argue that it's impossible, but some of my students at RIT try, because you can't tell someone it's impossible and then accept it. You have to prove them wrong, and then of course they realize there's so many components of color and structure that become difficult to use words to create a visual information of something that other can understand. Science photography makes things that are almost invisible or sometimes completely visible. So to be a good science photographer, you have to solve lots of problems. Every time I'm asked to photograph something, it's got unique problems that I have to resolve. And there is no off-the-shelf solution for most of the things. And I'm sure Felice will tell you in the same way. She's got to invent a way to do it. Constantly being challenged to innovate and do things that haven't been done before. And I have to really think about what I'm doing. I can't just pick up my camera and photograph, because there might be inherent things about the object that are required to be in the picture or not. So it's really important to understand the subject, the audience, the rationale, the equipment, the obstacles, and everything else, even before I commit to starting my session. And then there's the nature of, we want to believe that science is absolutely objective. 1 + 1 = 2. But I would argue there's not too much about science that's objective. It's a completely interpretive process. And depending on who you speak to, and depending on their background, and depending on their objectives, and blah, so much else, there's a huge subjective element to making pictures and doing experiments in science, no matter how we try to minimize variability. So making images in science becomes complicated because communicating is complicated. Science subjects by themselves do not make science pictures. So if I take a picture of a butterfly on the right and I ask a fine art photographer from North Carolina to take a picture of the butterfly, mine looks like the one on the right and his looks like the one on the left. Same type of lepidopteran, but completely different aesthetic and presentation. I don't think any science photographer would look at the left picture and say that's a science picture. It's beautiful. They're both different. How I photograph is hugely important. The 3 pictures on the left are me and the 2 pictures on the right are me, but the 3 pictures on the left would hardly ever be considered a science picture, but the 2 on the right would be considered possibly rhinoplasty pictures for a medical photographer. They have a certain standardization and alignments, and the others are completely interpretations of who I was on a given moment in time. Choosing backgrounds can be a very important element to the science picture. These 2 pictures came from a research collection in the Karolinska Institute Library in Stockholm, Sweden, made in the 1850s. Even back then, people who were photographing science understood the need to separate and isolate their objects. How we light is one of the singular most powerful and challenging components of science pictures. I'm sure most of you would realize this is a chicken egg, but maybe not. Each of those 4 pictures reveals a completely different characteristic of that egg. All would be considered technically correct. Which is the right one? I don't have an answer for you. It really depends on what you're trying to share. So each of them shares different information about the object. All too quickly in science, researchers are so focused on making a picture, pushing the button, they fail to slow down and think of what they're trying to reveal with that picture and to whom and for why. So lighting can share characteristics and expectations. These are seeds from a tree in my yard called a red bud. And in each picture, we see different information about the seed pod, the seed structures, how they're contained within. These are silver maple tree in my yard also. 8 different interpretations of one object lit in 8 different ways. All are correct, but all are different. Similarly, the muskrat skull. So, lighting is, it's kind of like the adjective to photography. How do you show and reveal things? So the light becomes the adjective to the noun. Science photography can expand time. Science photography shows how things change over time if practiced in a very standardized way. Science photography can stop time. In this particular picture taken in 1878 in California, there was a horse owner who believed that horses didn't have all 4 feet on the ground at one time. People didn't believe him. They said horses always have 1 foot on the ground at all of the time. And his series of pictures taken with 12 cameras discharged simultaneously revealed that the horse was airborne for a moment, and he won a significant amount of money from his friend who had to pay the price for the science fact. Science photography can see things outside of the range of human vision. So here we see a sunflower in visible light, and here we see a sunflower in ultraviolet light. This is a person who has put sunscreen on half of their face, photographed with ultraviolet, and we can begin to see why sunscreen blocks infrared from striking the skin and causing the melanin to get dark. Infrared, ultraviolet, changing from color to black and white, all are interpretive processes by the sensor, the photographer, the process. We need a scale in science photography. Without scale, science pictures don't tell truths. So it's very important for the viewer to know the size and relationship of things. Science photography can make behaviors visible to the viewer. So here we're looking at a type of crystal that I grew under my microscope. The left picture is just the structure of the crystal. By adding polarizers, I demonstrate a sort of birefringent property of how the objects and minerals within the crystals exhibit colors, which are diagnostic sometimes. In forensic scientists, we use colors often to determine what the object might be. By adding backgrounds, we can change a story. So science photographers are storytellers. They tell people who weren't there to see the science, what was going on, but the simple addition of flowers communicates being outdoors, and the simple addition of a white background communicates maybe we were in the laboratory. So the meaning of the picture can be changed simply by changing a background. So I started photography in 1972, as Charu said. Do you like my beads and my long hair and my rose-colored glasses and my film camera? The picture on the right was taken in February 2023, 50 years later, and I had just had completed displaying some science photography at of snowflakes. As soon as I bought my first camera in 72, I started taking pictures of anything that was in front of my camera. People, objects, friends had exhibitions. I was totally smitten by the process. In 1975, I went to the university to study pre-medical studies. My parents said, you can't be a photographer. You're not going to make any living money. You're going to have to go do something science. So they wanted me to study medicine, which I did, but I never lost my interest in photography along the way. I took histology and microscopy and cell genetics and anatomy and physiology, and the list goes on and on. I learned how to use a microscope on my own because there was no courses for this. It was great fun. Newspapers in the area where I went to school were interested in my pictures and got a cover on a weekend picture. I went to work in Syracuse as an advertising studio assistant for a little while, was losing money every week, not making money. So I had to leave that. I think this picture's hilarious, but I had to show it to you all. I was trying to be a rock star. And then I went back to the university in 1980, where I studied biomedical photography at RIT. I think I'm on time here. I have 4 minutes. So I learned how to photograph behaviors of light. I learned how to use a slit to make an image of what's called a peripheral portrait of a round object by moving film past a slit. I learned how to photograph things that were happening too fast to see, but I knew that they were happening, so developed a way to photograph a wasp in flight, stroboscopic motion analysis, and then went to work in Charleston, West Virginia as a medical photographer before moving to Detroit at Henry Ford Hospital, where I was the supervisor of photography there, working in operating rooms, photographing surgeries, laser surgery, all kinds of things that were going on in healthcare, cancer being removed, trying to, again, use standardized photographic practices. So it's very important. This person had high blood pressure. Before surgery, after surgery, the blood pressure had been managed. This poor woman, every time she'd raise her arms over her head, her eye would, so much pressure caused that orbit to want to push out of her head. Brain surgery, it's very powerful to be next to someone's human brain, or open heart surgery. This is a great picture where in 1982, cancer medicines were causing many of the persons who were administering them to develop skin cancer. You'll notice the person didn't have gloves. People didn't wear masks and gloves in the United States in 1982. AIDS was just becoming a disease in the United States. And so people who were administering chemotherapy agents were coming down with skin cancer. This picture was asked of me to demonstrate that when a syringe leaves a vial, there's a small aspirate, and that aspirate was the cause of getting skin cancer by all of the nurses who were administering chemotherapy. So a picture is worth 1,000 words. A picture can communicate things that we know happen, but we can't see happen. In 1986, I moved to RIT again, this time as a member of the faculty. Our school started in 1902. It's a very old school. At the time when I started, there was more than 1,000 students studying photography at RIT. The curriculum that I was in was the biomedical photographic communications. So I was dealing with things like shell casings and contact lenses and fossils and labeling and This is two Nasonia vitripens wasps having sex. The size of those insects was smaller than a rice grain. I put them in a small chamber. They were walking around rather than doing what they were supposed to do. You can't really ask insects to behave. They do what they're going to do. This is a female wasp ovipositing into a housefly egg. So very small. They're a good wasp because they're predatory. This is young Michael with lots of bellows and technical, very thin Michael, old Michael with, colored hair, not gray hair. And so what I thought we would do for just 2 or 3 minutes before I pass the baton to Felice, if we want to do, was I could share. Felice, you had mentioned you wanted to talk about a snowflake picture.
Very much. I'm very eager to find out how in the world you got this. How do you get it cold enough so this beautiful snowflake doesn't melt? Can you describe the setup for us?
Sure. So I work outside. That's a piece of black velvet material, and I catch the snowflakes on the velvet. The temperature in the US has to be between 15 and 25 for me to get the kind of crystals that I'm interested in photographing. I built a microscope out of used broken pieces of old microscopes from the 30s and 40s that function for me. So I catch the crystal on the velvet. It's like being a snowflake anthropologist. I'm looking to see what I have there. I look for the best crystal. I put a static charge on a needle. This is a small needle on the end of a pencil. So the static electricity makes the snowflake jump onto that needle. I move it to a microscope slide. I look at it and I use fiber optic lighting. I have about sometimes a minute to make this picture before the snowflake evaporates, which is a process called sublimation, or a process where it melts depending on the air temperature. So we can look at some more in a little while. I know that my 15 minutes just ended. I told you I'd be on time. So Felice, I'm going to pass the baton to you. And now it's your turn to amaze the audience.
Well, I'm not sure the audience will be amazed, but first of all, I have to tell you, I am honored to be in the presence and present with Michael. Michael is the science photographer. You have to understand, audience, that I am completely self-taught. I have no training in any photography. And I learn when I look at Michael's work, and it's something. The bottom line is, I don't think there is a profession in the United States, at least, of science photographers. Very few programs exist. That's a whole other conversation. But I want to just say I am honored to be with Michael, and I also want to thank Charu for being incredibly organized and spot on in how she's organized this whole thing. I'm very impressed. I wish we could work together more. So I'm going to share my screen, which basically I'm going to go right into images that kind of brought me to this world that I'm now part of. You're looking at my former life as an architectural and landscape photographer. I did graduate from Brooklyn College with a science degree, and that turns out to have come in very handy later on. But the bottom line is, I used to make photographs of architecture and landscape. This was in Picayune, Mississippi. This is the headquarters of PepsiCo, working in all kinds of weather, as you can see. Actually, it was fun for me to go back to these images, which, by the way, were all taken on film, and these are digitized because of a collection that MIT owns at this point of my stuff. But I really love doing this, but I was schlepping around the country with my cameras, trying to get the right images for magazines and for companies who hired me. Here is Osamu Noguchi's California scenario, taken in a particular situation, and I stayed there. And here's another of the same, taken in another lighting situation. Of course, photography is about light, isn't it? And so that's about learning and experience about how to light and what is the best way to represent it. And I agree Michael that there are no rules about this. We have to figure it out as we go along. Here's a winery in Napa, which I was kind of blown away by. I really had a wonderful time in this part of my life, traveling around. And here's this Napa winery taken in the winter, and here it is in the summer. Again, it's all about deciding when to make the image in architecture and landscape. It's might not be the situation in science, although we can argue that depending upon when you see something, that's a moment in time which can be different later on, and that's a whole other conversation we could have as well. And here, this is Ira Keller Fountain in Oregon. One of the parts of that kind of work was, do I want people in my picture, or do I want to just show it as it is? I'd actually prefer it without people, but that was a decision that I was able to make on my own. And it was a glorious time of my life. And what happened was, I was given this award at Harvard because of the work in architecture and landscape, and that changed my life. To get right to the point, I was taking a science class at Harvard because I missed science terribly. And then what happened was, one of the faculty that I was taking a class from, he was very visual. And I looked at his presentation, and I decided to go up to him after the presentation and asked him the audacious question of, can I come to his lab and see what's happening? And I did. I went to this guy's lab, and I saw that they were producing pictures like this. This was for a submission for a science magazine. What you're looking at is something called a self-assembled monolayer. It's a surface that's been treated so that this center, these lines are called hydrophobic, water fearing. And so when they drop drops of water on the surface, the water wetted the surface and stopped at the hydrophobic lines. This is his image, and this is about 4 millimeters across. And I said, you know what, maybe we could do better. It's an old story. This is in 1992, but I still tell it because it's relevant to what I'm still doing today. So I said, listen, oh, by the way, this is the way it was used in the article in Science. It was accepted to Science magazine. And I said, can we get a more interesting pattern? And what if we colored the water before we dropped it on the slide, this self-assembled monolayer? And so this is what we came up with. You could see, again, these are hydrophobic etchings. And I used blue and green dye because that's all they had. I wouldn't have selected it otherwise. And just as we know, just making slight adjustments changes the image. And I thought, okay, I had myself a potential cover. I even composed it so that the science label logo could be on top. But something very important happened. That's why I'm showing it to this day. The editors said they don't understand why the green is looking like 2 tones of green and 2 tones of blue. This is misinformational. It's not being, I'm not going to say it's not truthful, because it is truthful. That's the way the light responded to the dyes. But I went back because they wanted a simpler image. And eventually I came up with this. Again, these are fluorescing dyes, and I used UV light to excite the dye. This again is the same size. This is 4 millimeters across. This is a hydrophobic lines. And I'm going to show off a little bit because, yes, we got the cover. And what happened was, and this is all about luck. Yes, I did a good job, but I was in the right place at the right time. And whoever is talking, please mute yourself. And And what happened was George Whitesides, who turns out to be a very prominent chemist, I had no idea. He said, Felice, stay with this. You're doing something that not a lot of people are doing. And this led to another wonderful serendipitous moment. Is at the time I was collecting more images that I made for him, and was then got a position at MIT and decided to start playing around with other science labs. This is not my picture. This is a picture that somebody made of something called ferrofluid. I'm going to show you, the reason why I'm bringing this up is I want to show you the difference between just taking an ordinary image of something very interesting. And then this is the picture that I made. Why isn't it advancing, boys and girls? That's what I'm looking for. This is the image that I made of the same material. It's called, it's ferrofluid. What it is iron particles that are suspended in oil. I dropped this 3 centimeter across oil of ferrofluid on a glass slide. Under the glass slide is a yellow Post-it, very high tech. Under the yellow Post-it are 7 circular magnets. And so the iron particles are responding to the magnets. If you look carefully, you could see the window pane from which I lit it, just pure daylight. You can also see the remnants of a bad photographer, because here I am, without realizing it was in the picture until it was published in a book. But the difference between one and the other, I hope you see, makes a difference, because hopefully you want to ask questions about this photography. In this image, which was also at the very beginning of my MIT tenure, I was very lucky to be in Muji Buwendi's lab. You're looking at something called quantum dots, which in fact he just got a Nobel Prize for. This picture was taken 20 years ago, and it was one of the first images of the material. What you're seeing is cadmium selenide fluorescing at different wavelengths depending upon the size of the cadmium selenide. And this got a cover of JPhys Chem. They never put photographs on the cover, but the photograph was in fact the data. That's what Michael was talking about. Photographs are in fact data if properly taken and depicted. Here's a picture of the same material, but this was more for a cover of a brochure. But these are it was very exciting when Muji got the Nobel Prize, and everybody was celebrating. And his postdoc wrote to me and said, you know, Felice, you helped bring attention to it. Of course, it would have brought attention anyway, but it was one of these wonderful things that happened at MIT. At MIT, I have a blast. I'm learning about the science as I'm making pictures. This is Alice Nasto's picture of fabricated material that she made in order to study insulation. This is the picture she made. She gave me this, and this is the picture that I made of the identical thing. And I basically just folded it and lit it. Some of you might be interested in the fact that I couldn't get everything in focus, even though I was using a 105 macro lens. So I took a series of planes in focus and stitched it together. It's a very old technique called stacking, and it really works. And I just use software to stitch the images together. I use a scanner. I use microscopy. I use macro photography, but I also use a scanner, which blew me away when I first started using one. And I try to suggest to researchers to put stuff on your scanner. For example, the researcher gave me all these materials that he made. He wanted to try to get a cover of a journal. And so I took them all, put them on my scanner, started moving things around, And then we got the cover. This literally took 30 seconds. It's remarkable what you could. I edited the background. I digitally put it color in it, but it is absolutely remarkable what you could get. These are 1 centimeter across micro needles placed on my scanner. This is a euplectella. It's quite beautiful. The skeleton of the animal. I placed it on my scanner, set the DPI at a very high resolution. It's about a 7-8 inch long thing. And look what happened. This is the same image. I'm just zooming into the image. I'm not redoing it. Those fibers are 50 microns. You can do this on a scanner. Yes, it's a bit of cheating in a way because you don't work too hard, but you could put tomatoes on your scanner and get some absolutely remarkable products. And that a lot of the researchers are now using them for devices, and it really does work quite well. I talk about in a book that I'll tell you about in a second about using your phone. It is absolutely amazing what we could do with our phones these days. You can even get a cover of a magazine that I rarely would submit a phone image, but I didn't have to enlarge it. I composed it exactly for the cover, so it worked in this case. But we have to realize that there is an issue with file size. This is an image that I made with my phone of a petri dish with 4 sections of E. coli growing under different conditions. You must keep in mind that the file size is different from this image, which I made. Sorry, it's reversed. The first picture was the scanned image. The second is the file is a phone image, and you see that it is considerably smaller. And when you look at the details on both, you don't get the resolution. But that's changing. It is absolutely changing, and it's actually quite exciting what you could do with your phone these days. What I find myself doing now is I'm not an artist. I don't know how to draw at all, but what I can do is take bits of photographs and put them together to tell a story. For example, Don Sadoway gave me all this stuff to make some sort of image that replicates this drawing that is a metaphor for his liquid battery. And this is what I finally came up with. And if you go to my course, which I'll give you a taste of later in a moment, these are all bits and pieces of photographs that we put together to make an image. The same thing with this image. This is an image I just started with because I happen to like this plexiglass thing. And I layered it with graphene, digitally layered it, because I was going back and forth with the art director of Nature, and she and I came up with another way of creating an image that is something that you cannot see. It's a metaphor of an explanation. And eventually this is what we came up with the help of Nick Spencer, who's a wonderful art illustrator at Nature. Those are all pieces of images that I made that we put together. Here's another attempt at a cover. This is a picture that I made for a book that the researcher really liked the image, especially the background, which, by the way, the background is just a palette of watercolor. And as you see, the drop of water, in fact, is acting as a lens of the background. So you're actually seeing it in focus. But starting with this, I started stretching it. I took a scanned image of a syringe. I inserted the syringe in that image. I took images that they made of their fluorescing pancreatic cells. I inserted it in that drop and started playing around with it more. And eventually we got the cover, because it's a metaphor. What they're doing is alginating the pancreatic cells to protect them. So when they're infused into an animal, our macrophages do not eat them up. That's what that was about. But that was a story about how I put things together, which I'm starting to do a lot lately. But in the end, everything that I do absolutely maintains the scientific integrity. Whenever I do anything, if I do anything to a photograph, I absolutely inform the reader that I've done so. For example, Jerry Fink, a while ago, I made this image of his amazing yeast colony, and I was blown away about how gorgeous this was, but I wanted the reader to only look at the colony and really pay attention. So what I did was I digitally deleted the background. Because once again, I wanted you to pay attention to how these cells were growing with each other. What Jerry did not like the idea because he felt that I was deleting data. I maintain that in the caption, we could indicate that this was 5 centimeters across. He felt that if it were in the Petri dish, people would sort of know. It got the cover of Science, so he was pleased about this. But always, always I indicate when I'm doing something. When we look today at these extraordinary images from the James Webb Telescope, we don't know right off, or the world doesn't know that these are all digitally colored, that we have to, in my opinion, always indicate when what we're doing with science images is about being honest and letting people know about the process. What I found so interesting, and what you probably don't know, maybe you do, is that all these gorgeous star-like things are actually artifacts of the technique. That does not exist. That's about light diffracting off of these hexagonal mirrors. People don't know this. They see this as being part of the glory of the universe. And it's just a question of informing people about what is going on when we make pictures of science. Here's another example of something that I made years ago. These are Proteus colonies. And I didn't like the fact that the agar was cracking and it was distracting. I wanted you to only pay attention to these amazing patterns. And so what I did was I digitally deleted the cracks. When I showed, and by the way, when I submitted it, I absolutely indicated that I did this. When I showed it years ago, somebody suggested that, wait a second, maybe something is going on in those cracks. Did you have the right to do this? And this, of course, is an ongoing conversation. So what I'm going to leave you with this image is the fact that when you delete the outliers, if we could call those cracks outliers, we are deleting, what we're doing is we are changing the image, and by deleting it, we are affecting what we see. I ask the question all the time, and it's an ongoing issue. If we delete the outliers, are we deleting important information? It's a conversation that we must go through whenever anything is done, including colorizing images. It's a whole semester work of conversation. I talk about all this in my upcoming handbook. It's a teeny little book for $20. I'd love to, I'm unabashedly suggesting that you go pre-order it online at Amazon. It goes through all these very, very detailed, but very organized in a way that actually I was very pleased when I saw the book. You might also want to go to OpenCourseWare at MIT. I'm not teaching the class anymore, but there are 42 tutorials. Honestly, I think they're good. I think you could learn a lot. And you don't have to take the course. You could just go to OpenCourseWare and follow it through. That's it. I think, is that right? Is that all I have to say? Yes, that's all I have to say for now, unless we want to see a couple more.
That's
Yeah.
perfect. Thank you. Thank you so much, Felice, for sharing your journey and also showing us a lot of pictures. And it's very pertinent that you raised the point on, enhancement of the pictures. So, going slightly off the tangent from that, AI is the buzzword now. So what are your thoughts and what are Michael's thoughts on using AI for science photography? And there's also a question in the chat box that asks, how can artificial intelligence in science photography be used to open up new ways of seeing things in any other applications as well, apart from biomedical sciences or healthcare services? Michael, would you like to take that first?
Sure. I know we're at the beginning of this journey of AI. I've been talking to a few radiologists in the area that have been experimenting with radiology and mammograms and reading them, and the accuracy of the AI relative to the accuracy of the radiologist, and it's just not quite there yet. It has there's certain visual characteristics of the information provided, but I don't believe that it's very far in the future before it's going to be more accepted. I've played around a little bit with the generative filter in Photoshop, which is a new filter that can add information, but because most of its learning has come from outside of science, it didn't know what to do with cells and structures, and it created monster-looking things. So it wasn't really very good yet. The ethics of these things remain to be determined. The integrity of the
Can I
image
ask
that's
any
produced.
question?
I'm sorry?
Yes,
Can I
Vishal, you
ask
can put
any
your
questions?
question. You can put your question in the chat box and we'll take it up. Thank you.
Okay. If
So
there is
You can type your question in the chat box and we'll take it up, Vishal.
Felice, do you want to continue with the conversation about AI?
I'm nervous about it. I think it's absolutely, certainly in terms of medical challenges, I think it's very, very exciting. I think that even now, the machine can learn to read a cancer cell, even read it before the doctor, the physician gets to look at the X-rays or however the image they make. That is exciting, and I think the combination of the machine and the doctor will absolutely be an advancement as it continues. What I am nervous about is the deception. I will tell you honestly, I won't go back to it, but one of the pictures that I showed you, it was even the first picture of the landscape. I looked at it for the first time. Maybe I could just share. Do we have a moment to do? Oh, see, no, I closed my window, so forget it. Okay. It was the very first image. I looked at it. I said, "This is really lovely." But you know what? The sky isn't quite right. I'm getting more detailed about my own work. More critical, and so I generated the background to enhance the image. It was generating a new sky. I used the beta Photoshop, which is extraordinary, and sometimes it's not so extraordinary. I would never do this to a science image, and this is the question: how are we going to see if people are generating parts of their images, or if they're cleaning their images digitally? That's the scary part, and there's no answer at this point. There has to be some sort of oversight, and good luck on that. I think we're in trouble. I really do.
Well, it's a huge conversation, and as Felice indicated earlier, courses on ethics and image ethics and how to approach these subjects could be a semester-long course in a university to explore the boundaries of what's I've been involved as an expert witness a few times on are images truthful, and have images been contrived to create outcomes that aren't truthful. And the hard part of any of this is actually the intent of the researcher and the physician and the scientist. We can't measure intent. We can look at an image and read it, but we can't measure the intent of the person. And the question becomes, was deceit and being untruthful their objective or not? And so, we can marvel at images, and if they're used as illustrations to make points rather than to chronicle science. The boundaries are a little more fluid, but if we want to look at the image as the absolute facsimile of an object to be as truthful as the tools and environment will allow, then that's a very slippery slope to be on. If you do anything really outside of the boundaries of making an absolutely one-to-one accurate image of an object, which is actually impossible, but we do our best. We do
Yeah,
our
I
best.
want to, the other thing I want to say about that is that every camera is different. It already is, because they're digital. They already have a built-in algorithm to make it more pleasant. I mean, that's what the cameras manufacturers do. They just, what you see is not what you get. So ultimately, all images now are enhanced unless you manipulate your settings. And I'm not even sure, Michael, can we take a picture without the camera manipulating the photons?
Well, when I use my tools, I choose to turn off all of the automated features that the manufacturers have built into my camera. So I turn off sharpening, I turn
Right.
off color enhancement, I turn off noise reduction in the beginning. I use a color space that's accurate to what the eye might see rather than the sRGB, which is a compressed color space to try to be more truthful. I create the largest bit depth within the image that's possible. And then, as you shared, Sony and Canon and Nikon and Panasonic and all the other companies have sort of images, and digital cameras are grayscale. They're black and white. And the color that we get out of the camera is an algorithm written by the company that made that camera. So the color will be different. If I shot the same thing on 4 cameras, they'd all look just a little bit different. So it's a challenge, but it's not an obstacle. It's just a challenge, and to be aware.
To be aware, yeah.
Thank you. Thank you so much, Michael and Felice, for shedding light on that front. I would like to shift gears back to the basics now, because there are a couple of questions on how to go about becoming a science photographer. Now, you've been both on this journey, and this field is very niche and relatively, nascent in India. So what would be your career advice on how to, even start there?
Don't give up. Be persistent. Network. Volunteer. Shadow people, be persistent, be a pain in the behind, just constantly. What can I do to get in your lab? What can I do to work with you in healthcare? What can I do with you in biomedical engineering? Demonstrate a passion. Don't take no for an answer. If you have critics, go to someplace else and find another way in. I didn't have a straight path into the field. Everybody said, you're too creative. You're not going to make much of yourself in the science field because we don't need creative people. We need technical people. If I listened to them, I never would have done what I did. So you just have to be a little tough, not listen to your critics, continue to work hard and make yourself available, volunteer, go to meetings if there are, read everything you can read online. Practice. Those are my advices.
Lovely advice. With regard to the hard skills, what kind of courses can people, take to become science photographers? What kind of equipment they can start working with? Felice, would you like to answer that?
Well, I wanted to add something to the advice part before, if that's okay. I think the reason why I was able to find this niche is because I showed them what I could do. It's one thing to push yourself, invite yourself, but I literally, as I showed you, compared my picture to their picture, and it's a show and tell. It basically, they get it. And it wasn't easy when I got to MIT, by the way. A lot of people, what does this woman want, for goodness sake? But what I did do was I looked at the possibilities of the researchers who were clearly visual. You know, don't go to somebody who's a theoretician. Go to somebody who clearly needs pictures to demonstrate their science, and try to figure out a way that you would do it so that they would see you would do it better. You have to, unfortunately, it's the way it goes. You have to show that you could do it. That's an important part for me anyway. And the other part is I really do believe it's important to understand the science. You can't just go in as an artist. They're not interested in that. They want somebody who's going to show their baby pictures. I call them the baby pictures. But they're really, you know, I'm completely self-taught. Michael, luckily for us, teaches this. I don't think there are a lot of places that teach this, Michael, do that besides our RIT. There's illustration in some.
Yeah, there's medical illustration is a very big career in the US, and many universities teach medical illustration both at the undergraduate and master's level, but photographic programs in sciences are very small and far and few between. RIT has been in this space since the 60s. You know, we start out with fundamental photography in the first year. We teach them, but it's done with the scientific approach. So we have a course called phototechnology where we learn about lenses and optics and physics of image formation and color, and they learn about cameras and lenses. They learn about light. They learn about spectrums. They learn about variability and standard deviation. They learn how to measure and quantify and analyze results. In the second year, they learn about high-speed photography. They learn about optics. They learn about color reproduction, color management. They learn about the accuracy of devices. They learn how to profile. They learn about coding. They take a computer programming course. They have to take courses in sociology and reading and writing and history. And then in the upper divisions, they can focus. The industry in the US allows students with this background to go into the automotive industry, the image analysis industries. There's a lot of standard targets that are used in fields so that everybody knows that they're photographing in a correct and accepted way because the target that they have used communicates if the optics of the camera system are good or bad. You know, we can make, I would argue that no picture is truthful, actually. I would argue that there is no truth in photography. It's completely an interpretive process, but many of my science colleagues would say, well, that's not right, but you know, that's okay. That'd be a fun argument to have, because you know, where I focus, where I point my lens, what I choose to include and exclude is a very subjective thing, and so I could lie just by changing how I compose something. You know,
So I totally agree with you, Michael. I have a talk that I call representations are representations. They are not the thing. Photographs are representations. And it's hard for people to believe it because everyone's a photographer these days. So it's an issue.
We all have one of these.
You bet.
And I use my cell phone as much as anybody for photography, but not too much in science. I just value the optics. The optics of cell phone cameras are not on the par with the optics of other cameras at this point yet. But who knows?
It's getting there. At least the file size it's getting there.
Right. Yeah, very, very well put. Representation versus representation. I know that we're sort of nearing the end of the webinar, but I would just like to ask one last question. How do you go about communicating science to a general public via your photographs? So I understand that photographs go into medical journals or are for very specialized audience, say scientists, to elicit information. But when you have to explain science to a general audience who does not understand science, how do you go about it? Maybe you could just show a picture and, you know, as a fun exercise, the audience can find out what is sciencey behind that picture. And then what's the real story?
Well, I could share my screen if you like.
Yeah.
And quickly So does anyone, can we have someone guessing what this is?
Okay, if no one is, I'll take the opportunity. Looks like droplets on glass, but they're like very colored. That's my
Very
guess.
good. That
That's my
is
guess. Vapor.
your,
Someone says vapor. Michael, what is your guess? Condensation on glass. Okay.
Well, it's some sort of liquid, but what's in the background? It could be a postage stamp. It could be any number of things because I use the same trick when I do
Yeah,
Someone says
snowflakes.
fat droplets. Sorry. Yes, go ahead, Michael.
Oh, I just used all kinds of food boxes and plastic bags behind my snowflakes to make them colorful.
Ah, that's interesting. Any other guesses from our audience members? Droplets, but could be a colored background. Refrigerator fruit veggie basket. Drops raised from vapor from glass. Okay.
so
Yeah.
you're
Felice,
all
you can
right.
reveal.
You're all right. They're droplets of water, but it's not but. This is about taking a picture with my phone when I saw the science. And it was in about 30 seconds, it was all gone. I was sauteing colored peppers in a saute pan, and the cover was glass. And
Okay.
what you're seeing, you're not only seeing coalescence of the drops, but you're seeing an optical, magical optical issues, because they are in fact reflecting the color. The drops are reflecting the colors, and I show this because I'm working on a book for kids that science is everywhere, certainly in the kitchen, but it basically is about, you ask how do we speak to the public. I believe that if you make an image engaging, not just documentary. Let me see if I have another one. Here's another one. Let me just ask the audience, do they want to take a guess about what this is?
Whoa, this looks like I don't know, a wild guess. Potato.
A potato.
Yeah, that's what came to my mind. Any other
Okay.
wild guesses from the audience members? Yeah.
Michael, you want to give it a shot? I'm putting you on the spot.
Well, I see some scratches. It looks like some type of optical element in something, or a petri dish, but you have some polarized features going on.
Nope, no
I can't.
polarization. Yeah, it's actually the bottom of a copper pot, which has been oxidized because it was heated throughout time. But once again, the point of this is, for me, is to make it a guessing game, to get people to want to look. And then, the problem is science is so complicated, people are frightened. They're frightened to ask questions. I think one way to engage them is to make a picture that is attractive enough that they want to find out what it is that they're looking at. So I'll stop sharing. And I oh, did it stop?
Yeah, it
Okay.
did.
Okay. Yeah, I'm not, yeah, we'll leave it at that. Okay.
No, but those are like incredible visual stories, and it's very well in line with the theme that we started this conversation with, seeing science through lenses. So another piece of advice for aspiring science photographers is to make the audience see the science behind the pictures that they capture. So perfect. Thank you so much, Felice and Michael. Do you have any other thoughts to share with us before we close?
Someone asked if I had a little video of taking pictures through a snowflake. I don't know if
Yeah.
we can conclude with that, but
Yeah, I'd love it.
it's not, it was run on the internet, so it's not going to be super
Oh.
exciting, but
Oh, wow.
Wow.
Wonderful.
Yeah. Thank you for taking that question. All right, so it has been a great discussion, a great learning session, and I'm sure our attendees share the same sentiment. With this, I thank Felice and Michael for being so kind and taking out the time to deliver this wonderful webinar at 9 a.m. their time and 6:30 p.m. our time. We also thank the audience members for their active participation late in the evening on a Monday. And before we close, I would like to reiterate to our audience to not forget to submit their entries to our Science in Focus competition. The form closes on 27th October. Well then, thank you once again for joining us. Bye-bye. Take care.